Welcome to Neurosalience, the OHBM podcast.
Welcome to the Organization for Human Brain Mapping Neurosalience podcast. I'm Peter Bannattini, and here I discuss brain imaging and modeling with scientists from around the world. Today, we zoom in on Vancouver, British Columbia, to interview Dr. Todd Woodward, who's a professor in the Department of Psychiatry at the University of British Columbia and director of the UBC Brain Dynamics Laboratory. He's also the director of the Cognitive Neuroscience of Schizophrenia Laboratory at BC Mental Health and Addictions Research Institute in Vancouver. Dr. Woodward received his Ph.D. in Experimental Neuropsychology at the University of Victoria in 1999 and performed his postdoc at the Department of Psychology at UBC. Since 2003, he's moved up from research scientist to professor, all at the University of British Columbia. He's been working at the interface of processing methods and well-crafted experimental designs to probe the networks that may be disrupted in schizophrenia and other disorders. He and his team developed almost 2 decades ago a unique and elegant method known as Constrained Principal Component Analysis, or CPCA, which he's been applying successfully with many different tasks. He's also deeply interested in non-pharmaceutical interventions that help augment schizophrenia treatment, having developed a program called Metacognitive Training, MCT, which may allow those with schizophrenia to be able to begin to step back and begin to assess their own beliefs. This was such a wide-ranging conversation, which delved into the nuts and bolts of CPCA, as well as the potential future role that neuroimaging can play in better understanding and ultimately treating schizophrenia. I hope you enjoy it. And today my guest is Dr. Todd Woodward. He's a neuropsychologist, as you heard in the introduction, at the Department of Psychiatry, University of British Columbia in beautiful Vancouver. And well, thanks for joining us.
It is a pleasure. I really appreciate you having me on, and I'm a big fan of the podcast, so it's an honor for me to be your guest today. Thank you.
Well, thanks. And as I was mentioning to you, I'm always when people say they're a big fan or they like to listen to it, I'm always slightly surprised and very pleasantly honored that people listen. So hopefully I'm doing something right, and it's a lot of fun.
Absolutely. Very important service. Thank you.
So today, we're going to try to cover a lot of topics. And as I was also mentioning, one of the benefits of me doing this podcast is, I get to sort of really focus in on certain people, and some are very, very diverse. Some are methods, some are applications. Your focus is sort of this balance between method development and psychiatric applications, and you spending a lot of work on schizophrenia and in the context of that, working memory and other sort of cognitive tasks, and a lot of things. And you've been using for the past 20 years processing approach that we'll get into.
It's called fMRI CPCA.
fMRI CPCA.
Yeah.
Okay.
Constrained Principal Component Analysis.
So yeah, we'll get into that and sort of delve into how it's similar or different or unique and useful for the questions that you're asking. So, as I always like to start with a little bit of your background.
Right.
It looks like you've been spending all your life in the British Columbia area. And
well, I don't know if you know where Victoria is. Actually, west of Vancouver, on an island called Vancouver Island. That's the largest island on the west coast of North America. And there's a city there, Victoria. Is about 300,000, 250,000 people. And that's where I grew up and also did all of my university education.
Okay.
And so at that time, this was the 80s. And at that time, it was one of the top neuropsychology universities in North America. It still is a place where you can be registered as educated as a clinical neuropsychologist. So when I went to UVic, basically it's in my neighborhood where I grew up, and that was a natural thing to kind of go over there and start studying. And in the psychology department, they had some real leaders in neuropsychology, like Esther Strauss, Ottfried Spreen. There's a sort of a Bible of neuropsychology by Spreen and Strauss, and now Spreen, Strauss, and Hampson. When I entered UVic and found out, for some reason, neuropsychology was just the most interesting topic I'd ever heard. I knew I was interested in psychology, but when I found out, basically the Oliver Sacks kinds of stories where there's brain damage in this area and the person had some bizarre behavior, I was just fascinated by the whole thing. And then to happen to be in my own neighborhood where I grew up in the presence of people that understood how certain types of brain damage lead to certain types of aberrant behavior, I was completely fascinated and just threw myself right into study of neuropsychology. After doing my honors degree there, then I sort of took a couple of years off, and I did have scholarships to go to grad school, but it took me a few years to get there. I was traveling and things like that. And then ended up going back to University of Victoria for my master's and my Ph.D. just because of life circumstances kind of kept me there. At that point, I found out that I wanted to do only research. I'm actually not a clinical neuropsychologist or a clinical psychologist, but I'm very interested in neuropsychology. So I was able to take some neuropsychology classes, but they wouldn't let me register in some of them because I had to be a clinical student. So I didn't really know what to do, and I was disappointed, but Michael Hunter was the psychometrician or statistician in the department, and I started studying with him, and we were studying principal component analysis, factor analysis, all the different iterations of that, dementia reduction. So I started doing that for a few years. And clinical neuropsychology, as you and probably many of your listeners know, involves giving batteries of tests that are standardized and sort of interpreting the way people are spared and impaired on that battery of tests. So during that time, Daniel Bubb came from McGill, from the MNI, and was hired at the University of Victoria. Daniel Bubb is a clinical or cognitive neuropsychologist. So that sort of, and Daniel was from the British school where there was development of tests for the purpose of understanding specific types of brain damage. So now I found out that in order to study the brain, you didn't actually have to give standardized batteries of tests. You could just make up your own tests in order to investigate specific aspects of behavior that were of interest to the researcher. So that was, to me, what I needed. I didn't want to be told what types of tests I had to give, and I certainly didn't want to learn how to give all the instructions in a standardized way. So Daniel showed me that we can just develop our own tests. But the purpose of cognitive neuropsychology was to understand the cognitive architecture. It wasn't necessarily to understand that brain area and what it does, but we would try to use the way the brain had been damaged and develop tests with interesting control and experimental conditions to try to find out how the cognitive architecture is organized. And so really that was the goal. And Daniel was motivating me about different methodologies for doing that. And there's a lot of talk at the time about single case studies and whether they were valid. But then I graduated and ended up going across the water to the University of British Columbia at UBC. And at UBC in Vancouver, Peter Little was there. Peter Little, well, actually, when I was a student at University of Victoria, I had another student who was a friend who was studying schizophrenia. And she asked me to do a principal component analysis on the symptoms that psychiatrists collected data on about 50 subjects. And we did the principal component analysis on the symptoms, and they clustered perfectly into exactly what she predicted they would cluster into. Symptoms of schizophrenia basically cluster into hallucinations and delusions on one hand, negative symptoms on the other hand, like flat affect, reduced speech, reduced movement, and disorganization, which is derailment of speech and also inappropriate affect, laughing inappropriately, and things like that. And so those three clusters made me really curious about how it could be that hallucinations and delusion were correlated with each other, but not with negative symptoms. So I became interested in the brain underpinnings of the symptoms of schizophrenia. And anyway, in Vancouver, I applied to work with Peter Little, who you may remember. I don't know if you're familiar with Peter, but he was doing PET work very early on with Frisson and Frakowiak, and they actually did principal component analysis that's not that different than what I'm talking about today on PET data way back in the 90s. So he was in Vancouver. So he
Okay.
accepted me as a postdoc, and I was working on this schizophrenia. I do half my days at Riverview Hospital. Still have the mug right here,
Okay.
and maybe I'll take a sip of it. Where we had this a tertiary care hospital in British Columbia that has all the most serious patients there. And so I would go there and work on this cognitive neuropsychology investigations of delusions and hallucinations, which meant I could develop my own test and try to develop tests that would correlate with the severity of symptoms, which it's not the case if you just give sort of working memory task or an IQ task or, any type of traditional cognitive clinical neuropsychological task. It doesn't correlate. So I developed tasks that did correlate. So I was doing that at Riverview. And then at UBC, working with Peter on fMRI, and that involved using SPM because Peter came from the British School of developing the univariate type of model. That's how it all started with the fMRI. But then Christian Ruff, who was an undergraduate at that time, who came over from Germany to work with me, and Peter Graf, who's a cognitive psychologist at UBC, who was one of my supervisors besides Peter Little. We did a study on the Stroop effect, or actually it was task switching between word rooting and color naming with Stroop stimuli. And so he kind of did the analysis while I was over in the psychology department. He was in the psychiatry department. He finished the analysis on the phone, and I said, "What did you find?" He said, "I found the anterior cingulate." And I said, "Oh, okay, interesting." And I said, "What other areas did you find?" And he says, "Well, they don't do it that way. There's published the papers on one area." I'm like, "What?" Okay, so we published a paper on the anterior cingulate, but after that, I ended up on a flight from Atlanta. I think it was Neuroscience was in Atlanta, and I guess this must have been around early 2000s. Flying back, I read a paper by Yoshio Takani, who was working with my supervisor, Michael Hunter from the University of Victoria, on this method called constrained principal component analysis. And by reading that paper, that's my favorite paper. It's Hunter and Takani, Journal of Education and Behavioral Statistics. Not a very well-known paper, but I think it's my favorite paper of all that are sort of in my filing cabinet back there, and also now on my hard drive. But when I read that, I could see that would be a very effective way to analyze, depending on how you set up the matrices. Basically, the idea is that you have timing information on one side of the equation, and then you have all the bold signal on the other side of the equation. And so it's basically using, maybe I'll just talk about it now, right? So basically, back then what we did was, because I just came from the SPM lineage, I just took the design matrix the SPM gave us that has the synthetic hemodynamic response model in it. And I used that to split the bold signal, all of it at once, into what is predictable from that design matrix and what is not. So that is usually known as Y hat is the predictable part of the bold signal.
Yep.
And then Y, the error, is what's not predictable. So Y equals Y hat plus E. So we have now around, at that time, about 25,000 Y hats, but now if we use 2 by 2 by 2, we have around 250,000 Y hats. So that gets submitted to a principal component analysis. In the first paper we published, what we end up with is temporal information and spatial information. Once a principal component analysis is done on data like that, the right singular vector can be imposed. It can be overlaid on the brain image, and we get networks. But the temporal information, that information can be used to get the bold signal changes that are associated with the task and all task conditions. Actually, each subject.
Okay, so if I understand that's right, so this is you're referring. So as you mentioned, you're referring to this is before the FIR basis set sort of. This is now picking a canonical response function, and but this is the where you take the ICA or the principal components, and then you attempt to model them based on the timing with the canonical hemodynamic response function.
Well, it's interesting that you say that, because I think what you just said is kind of the opposite of what we actually do.
Okay.
But what you said is the way it's normally done, because normally what's done is the PCA on the bold signal.
Yeah.
Then the components are correlated with the timing information.
Right.
But what I'm saying, and the only difference, and it just surprises me that this slight difference seems to be really confusing to people is
Yeah.
you do the regression first. This
Okay.
is the basis of constrained principal component analysis. It's
Okay.
2 steps. You use multiple regression first to constrain the data.
Ah.
The constraints can be on the rows or on the columns. And it's just a
Okay.
very, very general model that almost all types of multivariate analysis are just a special case of it.
Well, that makes
Anyway,
the PCA simpler in that sense. If you do
exactly.
yeah,
I mean,
yeah,
this is
and
the
you,
thing that if we do the PCA on just the bold signal or the
yep,
ICA, I don't use ICA just because PCA is like one line of code and ICA is pages. So, I mean,
yeah,
I've always done PCA, but ICA could also be done. But if we do that on the bold signal, that's a very, very complex signal. And it can be during resting state, or it can be task state, movie state. All of them are complex,
yeah,
right? But
yes,
if we have timing information, which is why task-based fMRI, which I'm so disappointed to hear that people are giving up on it, because
yeah,
that's all I've ever done. Because of, like I told you, the training and developing tasks and the interest in the brain and how the tasks elicit activity in the brain. And so
yeah,
we can use that timing information to take out the variance in the bold signal, and it becomes so simple. We
yeah,
go from this very, very complex resting state or movie state or even task state pattern without the timing, and we have not only what cognitive operations are elicited by the task, but also all of the other ones that would be involved in trying to remember instructions, being stressed out of the scanner, all the other confounds like breathing, heart rate, all that. It's in there, so
yeah,
complex. And I'm very in awe and appreciative of all the people that are developing such amazing and intricate ways to analyze those data. But if we take out, if we have timing information, and that can be from eye movements, it could be from EEG signal, it could be
yep,
from any type of way you can be determined that something happened at a certain time. With a
yeah,
task, when we have that timing information, we can take out about somewhere between 5 and 15% of the variance in the bold signal and analyze that on its own. So we
yeah,
extract that first. And now
yeah,
all of a sudden, it just becomes amazingly simple.
yeah. So the key, I mean, right. I mean, and actually, you're right. I mean, I think that. Well, first, I don't think that the field is giving up on task. I think there's now becoming sort of a hybrid. I mean, we're getting into sort of naturalistic stimuli where we keep the task timing. We know the task timing, and we try to do intersubject correlation with specific task timing. But that said, the key is knowing the task timing. And I think I saw one of your papers where you were attempting to look at hallucinations, and that's tricky because it's like you have to know the timing of the hallucinations, which you can know it. Obviously, it's they report when they, but it's I can imagine that there's some variance
Oh, yeah.
in yeah.
Oh, yeah.
um,
Well, I mean, I can talk about that paper, and I will just after I talk about the finite impulse response model, because
yeah,
that's really important. And
yeah,
so I think it was Dara Minook that published a paper that used the finite impulse response model in a working memory paradigm. And it actually, with that, so anyway, the first paper was using that synthetic hemodynamic response model. And that was okay. But then when I read Dara's paper, I realized that a finite impulse response model would be much better, because now that 10% of variance that we take out of the bold signal doesn't have to match any specific shape. It
yeah,
only just says, tell me all the change in the bold signal that has happened due to this timing information, average over all the trials. And so it's now, because we're not matching a model, we can actually go back and find the hemodynamic response shapes. And the step, there's this very simple step, which I'll just say, it doesn't matter if anyone understands it, maybe somebody will, but it's very, very simple. And so first of all, the PCA on the Y hats, right, on that 10% of the variance will be optimized to task timing. But if you
okay,
do the PCA on the bold signal, those components will not be optimized to understand the task time. They're optimized to understand the variance in the bold signal period, right?
yeah,
Even if you rotate it, they're still not optimized. But this way, if you extract out the past timing predictable variance first, they're guaranteed to be optimized to the task first. And secondly, the component scores, so the component loadings are
yep,
superimposed on the brain, but the component scores can be correlated back with the design matrix. And
okay,
that tells you because the component scores tell us over the scans the activity and how it changes over time. It'll tell you which components are most active at certain time points that are already specified in the design matrix. So that's a
Depending on the
very
weighting.
quick explanation. But if we have a model in there that is the synthetic hemodynamic response model, we'll only be able to see matches to that. But if we have a finite impulse response model, when we correlate the component scores back with that finite impulse response model, we actually get the bold signal changes for every condition, for every subject that have been induced by that task, with no assumptions at all about the shape that we have to find. So that means that without any, all we've done is assume that the task timing gives us some important information. And that's the one assumption. And so that's somehow valid. Of course, the same networks that are involved in the task timing may also be involved in the error too, doing things like, what am I supposed to be doing? Of course, they're also but if we have only that task timing, we can have no assumption at all about the networks we might see or the regions of interest we have or the parcellations, none of that. Just use every single voxel in the brain. And we don't make any assumptions about the shape of the bold signal that's induced by the task, and where and in fact, if the assumed hemodynamic response shape, if it's a requirement to match that to find a network, you will not be able to find the naturally occurring brain networks. It's impossible that the model of the hemodynamic response shape or the bold signal changes would exactly match the naturally occurring one, even with temporal derivatives and that kind of thing.
Let me just ask one question here then. So, so I mean, obviously, if you don't make assumptions about the bold signal, I mean, you throw it out. You don't really throw out, but you reduce a little bit your sensitivity because you're not using this strict model. What people typically do is they convolve a bold model with the task timing, and then from there they can deconvolve it to get what's happening during the task itself pretty accurately. But so I when you say you're not making any assumptions about the hemodynamic response. So I think, from my looking at, tons of data, the hemodynamic response function doesn't really seem to vary that much, but maybe what you're saying is that the shape of the function, depending on the underlying neural activity, will show some differences. So that's the key thing.
We often hear things said like there's a different hemodynamic response shapes in different parts of the brain.
Right.
But I try not to even say the word hemodynamic response shape anymore, because that is just sort of a shape that's used by physicists that are trying to model how the bold signal changes depending on, all different cellular mechanisms, things like that. All I'm really talking about is the bold signal change that's been induced by the task.
Yeah. So why couldn't you? Yeah. Okay. So why couldn't you simply?
That changes depending on the brain area, because the different brain areas think different things, are responsible for different cognitive operations. So sometimes it's fast, sometimes it gets suppressed, sometimes it's long. And so if you use a finite impulse response model, you can observe that, which is completely lost when all we're doing is matching. So even if we sort of account for the same amount of variance, more or less, we still won't be able to actually observe the hemodynamic response changes, which is much more interesting to me than just looking at the neuroanatomy, because coming from the perspective of designing tasks, I want to know how my tasks push the bold signal around. And then with the different task conditions, I want to be able to see which networks were responding to those conditions, which weren't. Okay. So that's exactly what the method gives.
Yeah. So that's cool. I mean, that's actually the way, I mean, everyone sort of has their ingrained ways of thinking of it. I think of, sort of trying to explore, you have a signal, and I would think of it in terms of, if you can. So I like your approach. I mean, it's sort of like a bottom up approach. Another way of going about it is try to deconvolve the hemodynamic response function from the derived time course to get at the underlying neural activity. But it sounds like that's noisier than kind of what you're doing. So,
well, that's the thing. I mean, you can fit with an assumed model and temporal derivatives and try to get it back. But why would we do that when we can just look at it? We can just observe it. And that's turned out to be really, really important for our research program. And we spend much more time looking at the bold signal changes than we do looking at the neuroanatomy. So it turns out that if we do it this way, without these types of assumptions, without parcellating, taking out the task-related variance first, using a dimensional analysis, using a finite impulse response model in that way, that the same networks emerge for all tasks. There's
Yeah.
actually a set of networks that some of them overlap with default mode, but some are different.
Yeah. So you've been doing this for a long time. So I mean, how did this idea originate? And then, so you've certainly gotten probably feedback at meetings. Why? I mean, I haven't seen it around that much, and
Yeah.
so I'm kind of wondering, is there a misunderstanding, or is this some sort of just people are just siloed and want to, they're used to their packages and they do their thing. I mean, the software's out there. It's like, freely available. So,
Well, the
um,
software, you mean, I'm not running a software development company here. So sometimes it doesn't work that well. So people really kind of have to contact me and work with me a bit on it sometimes, usually, unless they want to code it themselves. So I think partly that and partly, yeah, tradition. And there's just been so many different lines of research. And let's face it, task hasn't really been the dominant area. I mean, it's been mostly resting state. And I mean, there's been times when task had its heyday, and maybe it's coming back now, like you mentioned. But I think that, and maybe the fact that I'm in a kind of isolated part of the world might have something to do with it. I'm not sure. I really don't know how to answer that, but it just amazes me that I can go. I think I went to human brain mapping in Glasgow and sat there for 5 days and didn't see one person talk about bold signal changes induced by a task. It's always just,
really,
yeah,
yeah.
it's always just
You're-
anatomy.
well,
Unless
that's
it's
interesting. Yeah,
EEG, then we see lots of, you know, curves, N4,
yeah,
P300, whatever. But for fMRI, even though we know we're studying, that's what we're studying, I think we've kind of left the hemodynamic response shape as just that. But
yeah.
it's not the hemodynamic response shape that's interesting. It's the bold signal changes induced by the task conditions that have
Right.
so much information.
I agree. I agree, and I think, I mean, there's many reasons. I think there's a big thing. I noticed in Glasgow as well that a big push to look at shared data sets, and shared data sets are very easy to pool big data sets with resting state. And I totally agree with you. I think that there should be, yeah, there's all kinds of variations. There's hybrid, combined event related and block design. There's naturalistic. There's now predictive modeling of various there's a whole range.
Mm-hmm.
and
Yeah.
and yeah.
Well, I mean, a lot of the time now when past fMRI data are analyzed, people sometimes don't even use the timing information. They'll analyze the task, data just like resting state would be analyzed by computing correlate, parcellating, computing correlation matrices, talk about. But like I'm saying, those resting state and task state, well, overlap. The task timing only accounts for 10%, so they're 90% overlapping anyway. But if we started to use the timing information to extract out the variance that's related to task timing first, maybe people would be able to confirm that we can find the same networks in multiple tasks and start to have a common framework where we can compare all the different tasks and the way the way the different networks activate over a whole range of tasks. We've probably looked at 60 or 70 in just my lab by
Yeah,
downloading data and also just our own data that we've collected over the years.
yeah,
But we could, as a field, be really understanding quite a bit about what these networks are doing by comparing how they activate or not in different task conditions over
yeah,
multiple different tasks. So
yeah. No,
yes,
I totally agree with that. And I think some people are trying to, like, I know that the Enigma consortium is trying to have a battery of tasks, and I do think that, yeah, we're getting there. I think that, we have, sort of a battery of networks. We have we're trying to create an ontology of sort of understanding cognitive states or tasks, and we're getting there. And I think that you're right. And it's kind of a natural dimensionality reduction in all this too. Who knows? I mean, like, it could be that the brain doing, many different things, it sort of reduces to some combination of a few networks that might,
well, I
might be
mean, let's
critical.
not forget that we're talking about fMRI. So we're talking about bold signal and how it configures into networks. And so it's obviously much, much simpler than trying to understand how 86 billion neurons form networks in different frequencies. So we're
Right.
lucky in the sense that bold signal is just much more accessible in terms of dimension reduction. And
Yeah.
actually, it can be great. Like I've been saying, things are greatly simplified when we can take out that percentage of variance. And I know it's kind of hard to believe, but because it's difficult to present the evidence to people for it, but the networks that can be derived from that percentage of variance are actually very specific. And we have very specific patterns that we're just starting to kind of release now. So to help us identify those networks when they emerge.
Well, maybe we should get it. So as far as getting to that a little bit, I mean, in general, right. The problem of, I've been, at the NIH, years ago, we had Danny Weinberger studying it, and this staff scientist, Joe Callicott, and other people, and there's many other people, and they always had, it was interesting because this was even before resting state. They looked at working memory, and they were trying to derive information. So maybe to back up a little bit, it's like, obviously there's many different types of schizophrenia and psychosis for that matter, and my suspicion actually is that it's all sort of subcortical processes that might be wrong, and then it's manifest in the cortex, but there's very clear behavioral differences and network differences that occur with working memory or whatever. So what's the goal as far as probing it in this way with these tasks?
Well, I actually wouldn't say that working memory. I started studying working memory because of schizophrenia. I think actually I kind of inherited it from the lab that I entered into, Peter Little's lab. They were kind of already doing that. So I'm not going to be able to answer a question about schizophrenia with the working memory, although I follow obviously Joe's work very closely, and that was inspirational for me. But what happened was I think I got the data, or I got the task. And the reason that became so interesting to me is because of the duration of the maintenance period. Because I'm looking at the the bold signal changes induced by the task. And we get changes in some networks and not others when there's an increased load, and changes in some networks and not others when there's increased duration of the maintaining period. And then the response period sort of comes at the end. So when we take out the task-related variance, do a dimensional analysis, all of those networks just emerge beautifully. And not only the anatomical depiction of the networks emerges, but also the bold signal changes induced by the task, which can be, like, for example, the network that's responsible for making reverse inference error by calling it the maintaining network, because that network is active in many tasks. It's not a working memory network, but that network, has an increase in activity for a higher load that gets even higher as the duration gets longer. But no other networks do that. Even the default mode network, which deactivates during the working memory trial, it does have higher activity for higher suppression or deactivity for higher load, but it doesn't get more deactive if the duration of the maintaining period is longer. So now we know that that's probably not really involved in maintaining the information in working memory, but the other one is. So that's how we can, that's why I started using the working memory task, or at least the Sternberg task, so much, is because it was so informative and helpful with developing the methodology, but really nothing at all. So, of course, I fit that into the literature, which was Joe Callicott's theory about inefficiency. And so we published, confirming that. But the reason I continued with it wasn't because I was more interested in inefficiency or frontal lobe or anything in working memory. It's because it's a great task for developing the fMRI CPCA methodology. And I get back to the hallucinations because that helped me decide how we would study hallucinations, because there's claims that hallucinations, superior temporal drivers, would be active because the areas that involve in hearing a voice would be the same areas that would involved in perceiving a voice. And so I said, okay, we had some data we shared data from around the world, from Australia and Netherlands, and we decided, okay, if we're really measuring hallucinations, let's bin them into short and long. And if we're actually picking up the hallucination, there should be a short hallucination and there should be a long hallucination. There should be a network that activates for a short period of time and for a long period of time, depending with the short or long hallucination. So that was sort of the criterion we put in for definite proof that we're measuring hallucinations. But maybe, like you've been mentioning, the people aren't that good at saying when they start and stop. So at least, even if we can't find short versus long, we should be able to see some network besides the button pressing. So we also, from the Melbourne data, had people listening to radio clips of radio speech.
Yeah.
And so those, even though they didn't design them to be short and long, we just listened to them, found the timing, and just bin those into short, medium, and long. So we did the analysis, looked at the bold signal changes induced by the task. Beautiful superior temporal gyrus network emerged for actually listening to the sound file. That
Yeah.
was perfectly short, medium, long.
Yes.
I mean, it couldn't have been any better. So
Yeah.
the data is good, but we go and look at the hallucinations. There is no short, medium, long. There's also no short, long. There's no bold signal induced changes that follow that pattern at all.
In the superior temporal gyrus, in the auditory cortex.
In anywhere, there
Yeah.
just isn't one. Okay, so
Huh.
we're not passing the hard test of
Yeah.
whether or not we're actually picking up hallucinations. So that's okay, because maybe people aren't that good at it. So what networks do we find? The response network only. Because we're pressing the button to say when the hallucinations came on.
So what do you think's happening? I saw one paper that you were in the title, at least it was mentioning silent hallucinations in some
Right.
sense. Where, what do you think?
Well,
So, and
I
also,
mean,
just to interject really quickly, I could have sworn I saw a paper, an old PET paper, with David Silberswig, with Verkowiak, I think as well, claiming to see either areas of the brain, or maybe superior temporal gyrus with PET, with hallucinations, but it could have been, obviously it was early, and who knows.
oh yeah.
Yeah.
Well, there's plenty of them saying that. And so problem number one, I'm trying to say something that is completely opposite to what the literature says. So that
Yeah.
paper got desk rejected from 6 journals.
Yeah, I mean, negative findings are always tough
Yeah,
because you could be
especially
like, "Oh, it's too noisy," or something, or whatever. You did something wrong.
right, did something wrong. I know exactly. So anyway, so the one question is, why does the literature say this? But the other question is, oh yeah, what's going on? Okay, so
Yeah,
what I think's going on. So Claire Humston and I just had a paper, an opinion piece selected for publication in Lancet Psychiatry, so it should be out within a week or so. I don't think it's quite out yet. And we argued that hallucinations are not. Now I know, at least from fMRI, I know that hallucinations are not the same as a perception, auditory perception, because I found the networks involved in auditory perception, they're very, very easy to find. Everybody who's ever analyzed any data for
yeah,
auditory perception, it's just perfect type of
simple.
superior temporal gyrus.
Yep.
But hallucinations, we're not picking that up. So it's not exact. fMRI, according to fMRI, somehow it's different. Exactly how it's different, I can't tell from fMRI as far as I can see. And so we think that hallucinations are more like beliefs. So beliefs that, even though people believe they're hearing a voice and they believe that the voice can come outside and they have that experience, but we think that the types of thoughts that happen outside of the hallucination are so similar to the types of thoughts that happen during the hallucination that the contrast that fMRI is all based on, right? This time versus this time, there was no difference,
Yeah.
except of course the response network.
Yeah, and just to clarify, I mean, it's very different than if you had people do, like, non-schizophrenic people doing mental imagery of, like, if they can do visualize something or imagine you hear something that actually could activate those areas. But with schizophrenics who have this illusion of hearing something, it's not like they're mentally imaging it in some sense. They're just experiencing the feeling of hearing it, I
It's not
guess.
a willful type of
Right.
thing. I think it is the when you willfully do something at a certain time, it's easy to pick up networks that are
Yeah.
involved in that. But when someone's pressing a button, when they have a certain type of thought, well, how different are those thoughts from when they weren't pressing the button?
Right.
According to this, not much at all.
Yeah. It could just be like more of a state of the brain that occurs. I'm sure that goes and waxes and wanes, where people, they hear something and they fill in their own. They can't control what they perceive in some sense, and so they sort of fill in. That's interesting. I mean, that's really, really interesting because, and yeah, no, I'm not surprised that, I mean, it's, one would think naively that they should, reactivate the whole system. But like you said, maybe the system itself doesn't need to be reactivated. It's just somewhere there's an internal representation of a reality that's not there, that they can't discern from reality. And that's just ongoing.
Yeah, exactly. I think that's probably what it is.
Yeah. And that actually touches on a little bit. And I really wanted to talk about this a little bit. I mean, to me, I think, like any disorder, it's potentially kind of a window into, our thought processes in some sense. And I mean, even among normals, we all every single person has this spectrum of, being perfectly rational and having beliefs that are not verified in evidence. And there's a whole spectrum. And obviously, if you have this too much, it's more of a disorder. And obviously there's reasons for that. We're still trying to figure out. And so it would be interesting, actually, right? I mean, I think you have studied a little bit in terms of the work on, trying to understand false beliefs or beliefs that, if you can't convince someone that they're wrong about something based on evidence or whatever.
Beliefs with weak supporting evidence, we called it.
Yeah, exactly. That's a
Covers
good
a
way to,
lot.
yes. And you actually had, though, I mean, so your non-imaging work that I have to mention. I mean, you've developed this app in which, obviously the state of psychiatry in general, I feel is it's hard because you have drugs that really haven't really been developed along a trajectory. I mean, the last drug for schizophrenia for it seems like it was developed years ago, and so that's sort of, we're not quite sure how they work, and if they're not quite sure if they work well, but there's also therapy, cognitive therapy, and it seems like you have this app now for I'm kind of curious how this works, and it's sort of basically, it's probably to augment drug effects, but to sort of, does it give feedback to those with schizophrenia about what's real versus not? It's like an extra person sort of talking to them. I mean, how does that work? I mean,
Yeah, so I like the way we're kind of tying this in with the history of how this all kind of happened. So I told you about how I was at UBC working on brain imaging with Peter Little and symptoms, and then at Riverview Hospital we were working on developing these tasks that would correlate with delusions and schizophrenia. And Stefan Moritz came from Hamburg, and we developed these tasks, and we found one that we developed is called bias against disconfirmatory evidence. So how willing are people to downrate a belief that they initially had? And it turns out that people with delusions, in the severity of the delusions is correlated, and I think it is correlated quite robustly, at least for the field of cognitive neuropsychiatry, with the severity of delusions. And so we developed that, and there's some other, like, how much evidence do you need to draw a firm conclusion? Theory. There was some work on theory of mind. How do you determine, by someone's facial expression, what might be on their mind? There's also in schizophrenia difficulty identifying emotions. So we kind of took all of those cognitive underpinnings of delusions and just basically took the task from our lab and from other people's labs around the world and put them into PowerPoint slides, and then sat people into a room and we just ran them through the task, basically. Like we said, what do you think's going on here? And it would be a picture of a guy, the dog beside his feet and the dog on the other side of the fence, and he's holding the fence like that. And it looks like he's playing with his neighbor's barking dog. There's a dog barking. And then you say, okay, here's what happened before that. And the dog's chasing him. And then before that, and the dog sort of, it's obviously that the man's been chased by the dog. He's not playing at all with the dog. So we show all these, that was one of the tasks that we developed. So by showing people that the idea, so metacognitive training is really, Stefan was the one who was this idea, and he's done most of the work, including translating into 33 languages. But it was at Riverview Hospital here in Vancouver that we worked together on sort of going back and forth from Germany to Vancouver. And when he'd come over for 6 months, we developed this training program called metacognitive training, which we don't talk to people about their own delusion specifically. You talk to them about how people reason, and you show them that, look, we don't understand the full situation right away all the time. In fact, usually we don't really have any idea what's going on until we get more information. So it's important to wait. And so people can easily apply that to much of their life. And then there's, there's 8 different modules and there's 2 versions. So people can go through and it's fun. You sit in a room, you talk about the scenarios. You don't talk at all about the person's own delusions or beliefs unless they sort of bring it up on their own. But they learn that, why, then they start to think, how come now I understand that I don't always understand the way things are most of the time in my life. So why am I so specifically sure about this one piece of information that everyone else keeps telling me is not true? It's not like it just cures them, but they develop new thinking tools that allow them to be less stressed out about it, because they can start to realize, oh, maybe this isn't true. I look at all those other scenarios and I learn now that we're not always right. Memories, for example, right? False memories. We run them through scenarios where you say, okay, remember all the things on this picture. And then later we take the picture away and put up your hand. And of course, people endorse some things that were not in the picture, some objects that were not in the picture. And then we show, look, even your memory's not accurate, so you can't be 100% sure of things. So anyway,
That's interesting.
it's been translated into 33 languages. There's been many meta-analyses showing the effect size of metacognitive training is similar to medication and cognitive behavioral therapy and cognitive remediation. And it's been published in top journals like JAMA Psychiatry.
Does it seem to imply that, we still don't, I can't even, I don't know the literature that well at all, but it seems like we still don't understand if, the neurochemical or even brain mechanism underpinnings of schizophrenia, or is it maybe just an extreme of a spectrum? It must be evolutionarily somehow selected for to be able to act and hold on to a belief if there's lack of evidence, and then being able to confirm it. But then somehow this is a disruption in that process of actually updating your beliefs. So, it seems that, is it just simply part of a spectrum, or do you think that there's a real neurochemical sort of problem, and this somehow is a behavioral intervention that might just help suppress that? Or
Yeah, I think mostly, you need medication and cognitive behavioral therapy and ideally metacognitive training. And notice that medication and cognitive behavioral therapy and metacognitive training all basically are focused on delusions and hallucinations, like the aspects of the disease that change quickly. But then there's just reduced IQ, there's reduced memory, there's sort of these cognitive impairments as well, which aren't impacted by medication or cognitive behavior therapy. But we have cognitive remediation for that, which also produces an effect size that's similar to all the other treatments. So we have a number of ways to treat schizophrenia besides medication. And so if we're talking about just something along spectrum and the medication affects that, then the neurotransmitters must be involved in that aspects of the illness. But at the same time, there's something very different about schizophrenia and beliefs without strong supporting evidence in healthy people. And that might be related to the cognitive impairments that are permanent. They're not changing over time the way the symptoms are. So there's kind of these state and trait aspects.
Okay. That's, it's really interesting. So before we get into some of your other brain imaging work briefly, it's like, what's your, right now, what's the most exciting thing as far as working on? I know you're doing many other things, but as far as, what is the sort of strategy? Like if you're thinking about future projects for addressing schizophrenia, what's the ultimate goal? To find something that's a biomarker for treatment, or a biomarker for looking at the progression of therapy, or just maybe for neuromodulation, maybe, finding the network that you could actually do TMS on or something. What do you think?
Well, the immediate goal, the one that's so achievable, is somehow making more accessible this whole range of treatment that aren't expensive. Schizophrenia will probably, we don't have the magic bullet that will make it go away, but we do have, like we've been talking about, so many different ways. And the metacognitive training can be also individual, and it can be the app like you talked about. And so there are a lot of tools we have. So I would like to see that made more accessible. The only way people usually get metacognitive training in Vancouver is through our own research program. So it would be nice if there was more psychology services available. So I try to promote that and try to, keep speaking around, not only Vancouver, but whenever I go to, I was just in Australia for 6 weeks and, talk quite a bit about down there. So that's achievable. But in terms of the next step, I did think was neuromodulation. And I have been working quite a bit with G10 system, which involves not only EEG data, but also inducing electrical activity. And my student Abhijit Chinchani has been working that for about 3 years. And that was the goal, because now that we talked about it, but some of I think I have a fairly good idea, at least from fMRI, which networks are involved in integrating disconfirming evidence. That comes from developing those tasks and, taking them into the scanner and finding one network, this network that we call reevaluation is more active when people are encountering disconfirming evidence than confirming. It's also correlated with the severity of delusions. It's also correlated with the same measure outside the scanner. So and this network that we call reevaluation is not only active during that type of task, but it's also active under a range of other conditions. But the thing about it, and it would never be detected with a normal model-shaped hemodynamic response model, or the traditional model, because this network peaks after response. So what we just said using the finite impulse response model, we just say, tell us which network to peak when. And this network, at first, I'm like, what's it doing way over there? Like everything's supposed to be over by the time people respond. But the reason is now that I see that it's involved in delusions and that it's involved in, integrating disconfirming evidence. When people are doing difficult tasks, like we just published a paper in Intelligence on the Ravens matrices task.
Yep.
So that's a difficult task. And this network reevaluation activates last. So first we have the networks that are involved in, doing the problem solving, then the response network, and then reevaluation network, because people are looking at the stimulus display that's still there and going over in their mind if they did it. If they did
the ability to like step back and sort of like so
And
what's the timing of that? How much later? Is it just like a couple seconds?
It's sort of the peak will be about 2 seconds later. I mean, it's not like it's close. It's quite distinct.
It's interesting.
And so anyway, now that we kind of understand that network and what it involves, we thought we would be able to try neuromodulation. Like maybe if you target that network, you can normalize it a bit. And then, when people go into cognitive behavioral therapy or metacognitive training, or maybe the medication will be more effective if you can just kind of lean things a bit towards a healthy state. But using the G10 to induce electrical currents has proven to be quite difficult. It's hard to control the electricity and exactly where it goes. So we never really got to the point of actually treating people. We're still experimenting with just reliably affecting behavior, whether it's eye movements or, performance or even just EEG measures. So, I mean, this is a 256 channel system. So we have 256 electrodes to induce electricity. And unfortunately, the amount that you can actually induce is not that much. Very, very small. So anyway, I don't think that's going to be an immediate path that will be very successful. So yeah, I'm sort of focused more on neuroimaging for moving forward. And treatment of schizophrenia, I think we've got a lot of options that aren't even used yet.
So, I mean, but even with neuroimaging, there's always neurofeedback. I mean, you could actually find the network that you want to enhance and, give sort of, real-time neurofeedback to see if they can actually enhance it. So TMS, you can't adjust the frequency of TMS such that it either stimulates the network rather than suppresses it, or I guess that's sort of tricky. And then, and obviously TDCS is it's very not accurate enough, I guess. But,
using TACS, but
TACS. Okay.
But there are lots of possibilities.
That's so cool. And so, what? Just out of curiosity, what network is this? I mean,
well, it's not one that's really well known. I don't know how to describe it. It's the activity in the frontal lobe is the most anterior of all the networks. In fact, one of the patterns that we use to identify it is we call it eyeball sitters. That's just within the lab. Maybe we'll start to publish these now, but it's directly above and right in the medial part of the eyeballs and the front part of the brain. Like you can see in the MRI scan, or when you superimpose the activity on that structural image, the eyeballs are kind of right there. So it's
oh, cool.
And it's a more lateral in the parietal cortex, but unfortunately, it's a bit difficult to describe the anatomy without a picture, and also it's also very, very difficult to try to say which network is most similar. But all of it's on GitHub and available for anyone to download. You can download these 11 networks and see in as much detail as is of interest to anybody what those networks look like. Yeah.
That's so cool. That's exciting I think there's something to that. All right. I mean, the whole idea of that maybe in schizophrenia, it's lacking that extra reevaluation sort of network that you sort of discovered through probing.
I'm sure that when people are doing that process of thinking, maybe I was wrong about that is the network that, at least that's what fMRI picks up. But obviously the neural activity is much more complex, but I'm quite confident that is the network that you would see when people are carrying out that type of process.
Interesting.
So then this gets into, like you were talking about, beliefs without strong supporting evidence, and people that are overly confident in their own beliefs that are potentially harmful. I think this is a really serious problem, and I would love to, see whether or not that network could be more active in people that are not following expert advice and things like that. So I'm not sure how to go about that, but I know that that's what our target is.
That's actually really cool. I think right. If you could actually have a network that you can modulate and cause people to be a little bit more reflective and a little bit more self-evaluation in terms of their own beliefs, and that would be awesome.
You go find them. and then
Yeah.
we'll do that. I'd like to come in here. You've got some beliefs that I think are not
Right. It's also
come on in
dangerous.
here.
I
We're
mean,
going to put some things on your brain, and we're going to modulate it so you think like us.
well, that's the thing. I mean, any time. I mean, that's always where the ethics come in. Anytime you, let's say, you could discover a network like that and you could modulate it. Is it ethical to modulate it then? In this case, that's a question that we'll have to answer eventually. The more powerful things get.
In
You're
the
right.
meantime, maybe we could just run multiple tasks and just try to become more confident that we know what the function of this network actually is.
It's also exciting that so Randy McIntosh, he does neuromodeling, and it seems like he just moved to your neck of the woods as well with Simon Fraser University.
Yeah.
So he's there. I mean, I think combining some dynamic modeling with this sort of thing could also be potentially informative.
Yeah, he's been really, since he's been here, it's been helpful to get UBC and SFU to get together more. He's been really doing a great job of making sure that neuroimagers from both sites are having lots of contact. And I have
Yeah.
talked to him about kind of mutual interests. And what I think is really interesting is that I think we have different goals. And my goal
Okay.
is probably not what most people's goal is with fMRI. So his goal seems, I don't want to speak for him, but it seems that he's trying to create a virtual brain. So in other words, if you could take eventually the long-term goal, you could have an individual person where you could have something that's close enough to their actual brain on a computer, and then you feed in their individual information about DTI and EEG and fMRI and all the brain measures you have, and then you kind of have some place where you could test, what about this treatment, what about that, what if we did this, what if we did that. So really, if you have any fMRI and data, what you're trying to do is say, what actual neural instantiation does this fMRI data imply? I've been doing the kind of work in the other end of the street there, where what I'm trying to do, I'm just trying to understand fMRI as well as possible, and I do that by interpreting the changes of the changes of the bold signal that are task induced, and to determine what the function of those networks is. So, I think how those networks and their functions are actually relating to the neural activity that is causing the bold signal changes is under debate. We don't know. And I think what would be great if we could decide on which networks we're interested in, then somehow using TMS lesion type models or just naturally occurring brain damage, and see whether a consistent sort of pattern is there. Maybe we can determine which nodes are actually essential for that operation and which aren't. So I'm not the idea of a virtual brain. Randy is just much more ambitious than I would be about that. I mean, that's the real challenge. But understanding the function of the brain networks, I think that we're not that far away from that. And this gets into reverse inference and that whole controversy. But if we have networks that replicate over multiple tasks, and we can see how the tasks are changing the activity in those networks, we're getting close, because, as Prozac has said, we kind of need a one-to-one mapping between a cognitive operation and the activity in fMRI to actually achieve reverse inference. But if we have the networks replicating over multiple tasks, how far away are we from being able to say what the function of that network is based on, say, hundreds or thousands of different tasks?
I think the inverse problem, right, is solved if you have enough overlapping tasks and you understand. And I think also Russ sort of is a big proponent of sort of like really trying to understand from a more reductionistic standpoint of what the tasks are. Working memory, it's like could be many things, and so I think part of the, at least, and better than I, the art of designing tasks is to try to make them as clean and precise as possible. And so even working memory is sort of like a combination of many different things. It could be a combination of just internal intention to your own thoughts and some, whatever that means.
I think that is a major component. But remember, I mean, I think that, first of all, saying a concept like working memory, even breaking that into subcomponents that are based on what cognitive psychologists might agree on, that's not necessarily what fMRI picks up. And it's not necessarily what neurons do either.
Yep.
So that's why, that's why I said I was making reverse inference error when I said that that network is involved in maintenance. And that's what we published at first, because we didn't know that network replicated in multiple tasks. But now we know, for example, that same network that's involved, and by the same, I don't mean like vaguely similar. I mean, eyeball thing and
Yeah.
specific patterns that we call crab claw or kite surfer that are literally identical in different
Yeah.
tasks. Like, for example, we just reanalyzed some data from Donna Addis's data that she published in the early 2000s that was on remembering, also imagining the future, imagining the past. The same network that's active during working memory, maintaining information, is also active remembering the past, imagining the future, imagining the past, something that didn't happen, but imagining. And so that network is actually more active in the imagining conditions than in the remembering conditions, for example.
Huh.
So
Huh.
so
Okay.
now how can we call that a working memory task? They're not trying to remember any information, they're just imagining the future. And there's other tasks, like there's a list of tasks I could talk about where that same network's active.
Yeah.
So now we forget about working memory. Forget about, that's not even going to help us understand the neurons do. It's not going to help us understand what fMRI is actually measuring. Just look and see that same network activating over multiple tasks, come up with something that fits. But unfortunately, that has been really difficult. And we've changed the names of the exact same anatomical configurations 3, 4,
Yeah.
5 times. And it's frustrating because every time I have a new co-author, they want to name it something else that fits in with their idea about what that general concept is. But we're getting closer, and it's not like you can really nail it down. It's
Yeah.
a cognitive mode. Every
Yeah.
network is involved in a certain cognitive mode, and to actually name that, you wouldn't name it by a cognitive psychology term. You just have to describe it in some way that is fMRI specific. So I think that's kind of the way it's moving forward to me. Not so much what the neuro correlation of that fMRI signal is, but what the function. This is a psychology perspective
Yep.
about the function. That's that nebulous thing between our behavior and the function, and what fMRI can really pick up. So
Yeah, so it reminds me a little bit of maybe what the goal of neurosynth is in some sense. I mean, you have neurosynth where, it's basically this massive database that just scrapes all the papers for the terms and finds the maps that correspond. And I'm sure there's a lot of noise in it. I've never heard of, some sort of, you can imagine some sort of, once again, sort of machine learning or AI algorithm sort of scraping that and then reducing all this information to, like you were saying, like the association between specific tasks and not networks themselves. But what Neurosynth does, it just shows the brain activation pattern. But the problem with that, of course, is that it's always they have unthreshold data, but it typically shows thresholds, and there's so many variables in how the experiment is done and processed. So it's probably noisy, but yeah,
I mean, what if we had for all tasks one set of networks that was actually retrieved for all tasks? Now you don't have the neurosynth problem anymore. One of the reasons why I think neurosynth is even needed is because using the model, the synthetic model of the bold change or the hemodynamic response involved with the task time, is that those will not be the same every time, just because the actual bold signal changes have to slightly miss.
yeah,
So it looks like there's a lot of different activations when actually I think underneath there's the same networks, but they're just slightly off each time. So you have
yeah,
neurosynth kind of averaging over all of these slightly missed. Sometimes they miss a lot. Like I already said, for the reevaluation network, you just
yeah,
completely miss it. We have a neurosynth right here. We could just look at the way the same networks are involved in every task, and then we don't really need to use that. Instead, we have an overarching set of
yeah,
cognitive modes that we
yeah.
can predict will be combined. Like the paper that Linda Chen and I just pre-published, that's got a whole, for the Sternberg task, we've actually put forward a set of 5 networks, as much anatomical detail as anyone would ever want, and also the predicted bold signal changes for a simple type of Sternberg task. It's all just
Oh.
laid out, and we should be able to do that for any type of task eventually.
Yeah, and I think that'd be really useful as far as, and I think right. There's many potential avenues where fMRI is going. Right. I mean, a big part of it, as you suggest, is sort of like resting state, and you parcelate the brain, and then you look at how the parcels are correlated, and that will only get you so far. But it may not pull out these more subtle networks, and like you said, it won't. It may not pull out the subtle timings of the interactions between the networks with specific tasks that you design. So there's still a big place for that.
Yeah, I
I
mean,
agree.
I just get nervous about, as soon as partialations happen right off the bat, people are forcing voxels onto the same network, or that if there's naturally a difference between them, that would be lost.
Yeah.
So
Yes.
it'd be difficult to find the specific types of patterns that I'm talking about.
Yeah, and certainly the simplification helps in some regards, but you're right; it throws away information in others. And I'm sure that you could probably parcelate the brain all the way down to, you know, on the individual voxel level, and even down to columns. If
Mhm. Oh
if
yeah,
that would be the most correct. Um,
right.
it would
Yeah,
be
I
fun
mean
to actually, yeah,
that's what I really think the future. I would love to see, just fMRI, because we've got, with these sort of 2 by 2 by 2 voxels, we have a disappointingly small number of networks. So with layer fMRI, or with better timing and more precise types of spatial information, there must be more networks, right? Like, and
oh
if we
yeah,
just allow, if we're using task and we take out that percentage variance and we use it for a model and allow the networks to configure on their own and to show us the bold signal changes that they have, then we can start to test hypotheses about more detailed networks. And so anyway, that's where I'd really like to see things go. But so far, I've sort of been bogged down in trying to just put forward these networks based on the whole brain. And I guess that's the other problem right now with layer fMRI, is that we're looking at usually just a small area, right?
yeah. Well, we're getting over that problem hopefully soon. I mean, with Bold, it can do it. Vaso. There's different versions of Vaso and Vapor and other things like that can do it, but there's still limitations of sensitivity and time and things like that. But yeah, it's getting there. It's
Yeah,
getting there. Yeah, and everyone needs a 7 Tesla too, I guess. To some do,
that's the way it'll
some
go.
argue. Yeah. So yeah, I could just keep on talking about this all night, but because I totally agree with you, I actually do. I completely agree but I'm still always thinking in terms of, you have these networks and people. I think that the nature of the field, and even I find myself doing this. I'm thinking, okay, well, let's say you take your reevaluation network, and let's try to find, let's parcelate that, and then see how it correlates with resting state or with tasks to other things, and try to probe that in some sense beyond just you found it with a task, and now let's see how it looks in other contexts. And yeah,
Well,
many things you can
yeah,
do.
that is something we're working on by using Yoshio's constrained principal component analysis. But we have been constraining only by the timing information so far, but we can also constrain by the spatial information. And so
Yeah.
we could just enter in each node of a network and try to
Mhm.
determine which combination of them is most important, because we can combine the column constraints and the row constraints together. That's the whole point
Yeah.
of what Yoshio did. And so we can
Yeah.
have the networks that are related to the task timing and the hypothesized networks, and also the other networks are related to the not or orthogonal to the hypothesized network, but still related to task timing,
Yeah.
and
Yeah.
ones that are related to the proposed networks and not are orthogonal to task timing as well.
Yeah.
But
Yeah.
that's just, it's so difficult just using the row constraints on their own that we've only Megan Rose, my PhD student, published using the anatomical constraints, but that's only pre-published. It's not actually hasn't been submitted to a journal yet. So
Okay. Well, that's cool. That's cool. So there's so many other things also. I mean, we're probably coming up a little bit on time, but there's so many other things that you've done in terms of looking at lexical decision making, pain perception, as we briefly mentioned, looking at evidence integration and delusions, and also right the altered network balance between default mode network and the frontal network in terms of evaluating things versus self reflection. You've done a lot of work, and every one of these is a conversation in itself. But I just want to maybe in sort of towards the conclusion of this, just ask you. I mean, you've already suggested as to what you might be interested in doing as far as further trying to probe the nature of the relationship between behaviors and these more subtle networks, and sort of build up this library in some sense of these relations, so you can actually get a more nuanced view. What do you think, as far as this is concerned? I mean, maybe someday we'll figure out what the mechanism is involved, and maybe it might involve neuromodulation. But do you think that things like, I mean, where do you think, this is a big, broad question. Like, where do you think the field of trying to understand mental illness and trying to treat it? What do you think the best hope is? I mean, do you think that we'll discover better drugs, or do you think that it's better through more nuanced understanding of the networks, we'll develop neuromodulation probes, or some combination of everything with therapy will further help things? I mean, so what do you think the future is? I guess.
that is a tough one. I mean, I sort of tried to answer that by, saying that I just wish we would apply what we already have, because we have so much that's partially effective.
Yeah.
But I 3 years ago, I would have said that the thing that is missing is brain stimulation, neuromodulation. And I still kind of want to have hope that that is the next step, because we don't even understand very well at all the brain networks that underlie mental health issues. And if we did, if we understood them well enough, then if we also felt confident that we could adjust them in some controlled way, we should be able to help that way. But it's just when you try to do it, things are just not. I think that is a fact, that it's potentially is the next big thing for treatment of mental health. We have to understand so much more about the brain, and not just how the bold signal flows, but how the neural, when we talked about hallucinations, I talked about bold signal. We can't detect with bold signal, but of course there's neural activity that's different when people are hallucinating compared to when they're not, even if fMRI can't pick it up. So, if fMRI can't help, could EEG, MEG, is there some way we can actually measure reliably those brain networks? And if we could have a very good idea about where it is, then we just need to be able to have confidence we can impact them with TACS or TDCS or whatever, or TMS, so I would still hope that is the future. And, maybe you have to think like Elon Musk. It's easy.
Yeah, with his neural lace, I guess, and
Neuralink, yeah.
Neuralink as well.
Replaced a whole bunch of the skull, and
that's
promoting that or saying that is right. I'm just saying that, sometimes when we're sort of, our hands are dirty in these fields, it just seems like it's very difficult. And maybe it isn't, but maybe there'll be a breakthrough. I don't know. I hope so.
Yeah, but also you have people like our current director of NIMH, Josh Gordon, is very focused on a very different temporal and spatial scale, looking at the molecular, the neural circuitry. The answer might be at that scale in some sense. Who knows? But there's a lot of work to be done. All right. Well, this has been great. So there's so much more. I always, especially with conversations like this, I'm always sort of regretfully, have to wrap it up, because there's so much more to talk about. It's so much. I think that the question of applying neuroimaging, neuromodulation to understand and help mental health is, I think, probably the biggest, most important use of neuroimaging. And so,
Agree.
and it's a long range goal, and the problem is messy, but it's worth doing. So, and it's been a pleasure talking with you. And
Thank you so much for having me. It's been a pleasure too.
Well, thank you. I wish you the best. All right. Well,
you too.
Thanks.
Neurosalience is brought to you by Organization for Human Brain Mapping. This week's episode is produced by Xiqian Michelle Li and Ömer Faruk Gülban.